Okay, so we've been talking a lot about signaling molecules, neurotransmitters, and hormones, but what's the point of all of this activity? That is to say, a neurotransmitter is going to bind to a downstream cell, but what is that going to elicit? One thing it's going to elicit is muscle contraction. So let's talk about muscles. We have three broad categories of muscles. We have skeletal muscle, and those are the muscles that are attached directly to bone. Most skeletal muscle is voluntary. One exception to that is the diaphragm. The diaphragm can be modified voluntarily, but by and large, the diaphragm, which is a muscle of respiration, contracts and relaxes involuntarily. It's part of our autonomic nervous system. But the majority of our skeletal muscle, or let me rephrase this, all our skeletal muscle is bound directly to bone, hence the term skeletal muscle. We also have muscle of the heart, which is known as the myocardium. We'll talk about that in detail when we get to the cardiovascular system. And to be clear, the myocardium and skeletal muscle are very, very, very similar. And one thing I want to point out, the prefix myo refers to muscle. Cardium refers to heart, so that's the heart muscle. One may talk about muscle cells. They may say myocytes. That is merely saying a muscle cell. So we have skeletal muscle, we have heart muscle known as the myocardium, and the third category of muscle is smooth muscle. And we find smooth muscle lining tracts like the GI tract, so it's lining the stomach, the small intestine, large intestine, the esophagus, which is superior to the stomach. It's lining tubes such as our blood vessels. So here's an example of a blood vessel. This is the lumen that blood flows through. Lining the blood vessel is endothelium, which is simple squamous epithelial tissue, not muscle. But in purple here, surrounding the lumen or circumventing the lumen is smooth muscle. And contracting and relaxing that smooth muscle blood vessels causes vasodilation and vasoconstriction. And we'll talk about that when we get to the cardiovascular system. If you remember talking about the uterus, the myometrium is smooth muscle. So there's a whole host of smooth muscle throughout the body. If it is not attached to bone, like skeletal muscle, and if it's not attached to the heart or a component of the heart, it is smooth muscle. So there is arguably more smooth muscle in the body than any of the other two types of muscle. So we're going to start our discussion with skeletal muscle. It is really kind of the basis for an introductory discussion on muscle. And once again, the myocardium or the heart muscle is very similar to skeletal. So we need to talk about some nomenclature before we get into the physiology, because there's specialized terms within skeletal muscle. Don't ask me why, but there is. One term that I don't have written up here is muscle fiber. Muscle fiber is often a term one uses when they're talking about skeletal muscle, because skeletal muscle tends to be elongated and looks like a fiber. So it can be complicated in the sense that we don't want to get confused with a nerve fiber or, let's say, a collagen fiber. To be clear, when I say muscle fiber, I am specifically talking about a skeletal muscle cell. Okay, so some of the special wording or nomenclature, skeletal muscle fiber that we see here. The plasma membrane or cell membrane of a skeletal muscle fiber is known as the sarcolemma. Sarcolemma and plasma membrane mean the same thing with regards to muscle cells. The cytoplasm of a muscle fiber is the sarcoplasm, and there is an organelle within skeletal muscle fibers known as the endoplasmic reticulum, specifically the smooth endoplasmic reticulum that is found in other cells in the body, not just skeletal muscle fibers. But the smooth endoplasmic reticulum in skeletal muscle fibers is referred to as the sarcoplasmic reticulum, and the sarcoplasmic reticulum stores calcium. And the last feature I want to mention here, other than the overall structures that make up the skeletal muscle fiber, is the T-tubule, and that's what we see here. So to be clear, what is happening here is the sarcolemma dips or invaginates in towards the center of the cell. So if I'm looking right here, this is the extracellular fluid, that's intracellular fluid. This is extracellular fluid, intracellular fluid. And right here, that is extracellular fluid. This is outside the cell because the plasma membrane or T-tubule is dipping in here. And that's merely to allow these other proteins to be in closer proximity to each other. If this protein right here, which is known as the DHP receptor, were up here on the sarcolemma, it would be too far from the sarcoplasmic reticulum to elicit any sort of response from the DHP receptor. So we have the sarcoplasmic reticulum that stores calcium. We have the DHP receptor, which I'll talk about that in detail. Right here, we have a ligand-gated sodium channel, specifically a cholinergic channel. It's a nicotinic channel because acetylcholine will bind to this channel and open it. Once again, it's a ligand-gated sodium channel. This is a somatic motor neuron. Certainly a somatic motor neuron is not part of the skeletal muscle fiber, but it is necessary to excite this cell. That is to say, to cause this cell to depolarize. In this state, we can see the negative charge on the interior, so this cell is at rest and there's no activity going on. The last thing I want to say about this is there is a super high concentration of calcium in the sarcoplasmic reticulum and a low concentration of calcium within the sarcoplasm. One thing that needs to happen for skeletal muscle fibers to contract, and contracting a muscle is shortening of muscle, and that shortening of muscle generates force, allowing us to do work using a barbell, dumbbell, picking up a trash can, whatever it is. Contracting muscle generates force, and one of the first things that needs to happen to generate force is to release calcium from the sarcoplasmic reticulum. Here we see the gate is closed. That gate is known as the ryanodine channel. So, no activity going on here. Here we see the somatic motor neuron firing. Why is the somatic motor neuron firing or depolarizing? This is all voluntary because skeletal muscle, by and large, is voluntary unless there's some sort of twitch or spasm that's going on, or a cramp. But intentionally, one is deciding to do something with their skeleton, whether that's walk, run, lift up a barbell, chop up some garlic, whatever it is. This depolarizes, releasing acetylcholine. That acetylcholine, when it binds to this nicotinic receptor channel, opens up this channel. Then we're going to get the influx of sodium. Sodium's going to move down its electrochemical gradients and depolarize this cell. And I'm just showing the depolarization right here. To be clear, for this depolarization to spread from this channel to this DHP receptor, which I see here, which, by the way, it's a different shape than we see here. This movement or depolarization event that spreads along the sarcolemma is due to voltage-gated sodium channels. So these channels are exactly like the channels we see peppered along the axon of a neuron. That is to say, like a row of dominoes falling. These are voltage-gated sodium channels that spread or propagate this depolarization from this channel to the DHP receptor. The DHP receptor is a voltage-sensitive protein. It is not a channel, but it is a protein that changes shape, undergoes a conformational change when there's a depolarization event. So we just went from a circle to this skinny little rectangle right here. One thing I failed to point out is there is a chain or a protein that is physically linked to the ryanodine channel. So this chain or protein fiber goes from the DHP receptor to the ryanodine channel. And when the DHP receptor undergoes this conformational change, this chain pulls on the ryanodine channel and opens up that channel. So here we can see the channel closed. Here the channel is open, merely because the DHP receptor underwent this conformational change and it pulled on the gate, pulled on the ryanodine channel to allow calcium to leave the sarcoplasmic reticulum. Now, why is calcium leaving the sarcoplasmic reticulum? Just because it's moving down its chemical gradient from an area of high concentration in the sarcoplasmic reticulum into the sarcoplasm. Now, that's the basics or the initiation of muscle contraction. We have yet to have muscle contraction, but all of this needs to happen even before we think about muscle contraction. One thing I want to point out are the pumps. Now, why do we need pumps? Because pumps restore gradients. Sodium is moving into the cell, so that gradient needs to be restored. There's a repolarization event I haven't even shown here, but keep in mind, anytime a cell depolarizes, it must repolarize, and that's going to be due to potassium efflux out of the cell. And certainly calcium needs to be put back where it came from into the sarcoplasmic reticulum. So to restore sodium's gradient and potassium's gradient, I should probably say to maintain, because restore is suggesting we lost the gradient, which we probably didn't. But to maintain sodium and potassium's gradient, there are going to be sodium potassium pumps within the skeletal muscle fiber to maintain sodium and potassium's gradient, pumping 3 sodium out of the cell against its gradient and 2 potassium into the cell against its gradient. 3 sodium out, 2 potassium in. We also have what's known as a calcium pump that pumps calcium back into the sarcoplasmic reticulum. Keep in mind, I'm going to put the calcium pump right here. This gate is going to be open just for a fleeting moment, like a lot of the gates I talk about. But we need to get calcium back in here against its gradient. So calcium is going to be pumped back into that organelle, into the sarcoplasmic reticulum against its gradient. This calcium pump is also known as the calcium ATPase. It utilizes ATP, it hydrolyzes ATP to liberate energy, and that energy is used to push calcium against its gradient back into the sarcoplasmic reticulum. So those are the pumps of interest specific to skeletal muscle fibers. So let's take a closer look at some of these contractile proteins that actually allow for muscle contraction. Let's take a look at these contractile proteins. Right here, these are fibers, muscle fibers, protein fibers within the skeletal muscle cell. So please just imagine this is within the skeletal muscle cell. These squiggly lines on the ends of these fibers are known as the Z-discs. And when these muscle fibers contract or shorten, they're pulling these Z-discs closer to each other. This whole structure between the Z-discs is what's known as a sarcomere. And to put all of this in perspective, let's just look at a key here for all the structures we're looking at. So the red circle, the individual red circle, represents what's known as G-actin. You see a bunch of red circles here in this. There are 2 strands of G-actin, and the G-actins make up what's known as the thin filament. So there's a thick filament and thin filament in skeletal muscle and in cardiac muscle, and those are the contractile proteins. So we have the thin filament, which is composed of G-actin and F-actin. F-actin is merely 2 different strands of G-actin molecules. G-actins are the monomeric subunit. In the black parts of the G-actin molecule, which we see along F-actin, is a binding site for the myosin head. Here we see the myosin molecule. It makes up the thick filament of muscle, and attached to the coming off the myosin molecule are these myosin heads right here. So we have 3 heads here, 3 heads right here. This green line is representing tropomyosin, and this blue squarish looking thing is troponin. Tropomyosin and troponin are regulatory proteins. They, with their activity, are going to determine whether or not the shortening of the muscle fibers can happen. To be clear, the contractile muscle fibers are the thick filament and thin filament, are the thick filament and thin filament. Thick is composed of myosin molecules, thin filaments composed of G-actin and F-actin. So let's take a closer look at this. Here we have the thick filament in purple. We have thin filaments above it and below it. The space between 2 adjacent thin filaments is known as the H zone. So from here to here is the H zone, from here to here is the H zone. So during muscle contraction, what happens is the thick filament, the myosin molecules, grab the thin filament and they pull the thin filament closer to each other. That is to say, these myosin heads are going to grab the thin filament and move them closer to each other diminishing the size or width of that H zone. So the H zone, which is this right here, during muscle contraction, that H zone gets smaller, and that's merely because the thick filaments with the myosin heads grab the thin filament and move those thin filaments closer to each other. The thick filament is not really moving other than the myosin heads, which are moving like this. So once again, thick filament in purple, thin filament in red. Now, in the setup we have it here, in green, once again, that's tropomyosin. That is a regulatory protein. The reason muscle contraction or shortening cannot occur in this situation is because tropomyosin is blocking the binding site for the myosin heads. The tropomyosin is blocking the black binding site, which is the binding site for these myosin heads. There's no way for the myosin heads, either up here or down here, to be able to grab the thin filament because tropomyosin is in the way. And it's intended to be that way to ensure muscle contraction does not occur when we don't want it to occur. The other regulatory protein we see here is troponin. Troponin is physically linked and attached to tropomyosin. As long as nothing is bound to troponin, muscle contraction cannot occur. For muscle contraction to occur, we need tropomyosin to move out of the way. And that's what we have down here. We actually see the shortening of the muscle fiber. The H zone, which is right here and right here, is much smaller than it is up here. And that happened because the myosin heads grabbed the thin filament and pulled those thin filaments closer to each other. I always imagine it like sliding doors. You grab handles on a sliding doors and you pull those doors closer to each other. That's what the thick filament does to the thin filament. Goes up and grabs the thin filament and pulls those filaments closer to each other. And we'll talk about the details of the sliding of the filaments in a moment here. But just so we're all clear, the reason why tropomyosin is no longer blocking the binding site is because calcium has bound to troponin. When calcium binds to troponin, troponin undergoes a conformational change. I've tried to show that a little bit on the difference in shape between here and here. I wasn't as successful here and here. But please understand, when calcium binds to troponin, troponin undergoes a conformational change, and that conformational change takes tropomyosin and pulls it out of the way, liberating the binding site for the myosin head. The myosin head then grabs a thin filament. Specifically, the myosin head grabs individual G-actin molecules and fires, and then we get shortening of the muscle. Okay, so how do these filaments actually slide? And what we are going to use to describe this is what's known as the sliding filament theory. It is the predominant suggestion on how this all happens. Similar things happen within cells. For example, in neurons, there's proteins that actually step upon each other to transport organelles, say like secretory vesicles, from the cell body down to the axon terminals. So once again, the sliding filament theory, this is a cycle, if you will. So we could start anywhere within this cycle. One thing I want to point out, this is broken down into very smaller components. So this right here is representing the thin filament. I realize I only have one strand of F-actin, individual G-actins there, and I only have one little aspect of the thick filament or the myosin molecule with just one myosin head. So I'm going to go through this once, I'll go through it again and break it down into its smaller components. So the first stage I'm going to start out with right here, which I have labeled detachment, but at this stage don't worry about that because attachment hasn't even happened. But what I want you to focus on right here is that the myosin head is not bound to the thin filament. And it's actually, the myosin head is in this fired position, somewhat of at a 90-degree angle right here. But most importantly, the myosin head is not touching the thin filament. And one thing to understand when we're talking about the sliding filament theory is that this assumes that the muscle fiber has been depolarized and calcium has been released from the sarcoplasmic reticulum, and most importantly, calcium is bound to troponin, and troponin is moved up and out of the way, liberating the binding site for the myosin head on the G-actin molecules or on the thin filament. So we don't even see troponin or tropomyosin in this depiction right here, but just understand and assume that tropomyosin has been moved out of the way because calcium is bound to troponin. So one thing you may notice here is that ATP is bound to the myosin head. So to move from this stage to this stage is the result of ATP hydrolysis, which means ATP has been split into one molecule of ADP and one single inorganic phosphate, which I've neglected to write in here, so let's do that right now. Okay, so once again, we've got the one ADP and the inorganic phosphate bound to the myosin head. One thing to keep in mind with the myosin molecule, and specifically with the myosin head, this is a contractile protein that grabs both thin filaments and moves them closer to each other, decreasing that H zone. And to be clear, all of this is just one side. So I'm showing one side here, but keep in mind this is happening on both sides and even at the bottom of the myosin head, grabbing the thin filaments below the thick filament. But what I was trying to say is that the myosin head, in addition to being a contractile protein, is an enzyme. It's known as myosin ATPase. And because it's an enzyme and it's ATPase, it's going to hydrolyze or break down ATP, and that's what happened from here to here. Because the phosphate ion is still bound closely to the myosin head, no energy has been released yet, maybe a little bit, but the main thing that happens during ATP hydrolysis from this stage to that stage is that myosin head goes back to vertical and grabs the thin filament. Here, it's not grabbing the thin filament. And one thing I've shown here on the tip of the myosin head that I have outlined in black is the part of the myosin head that has a great affinity for the thin filament. That is to say, if it's oriented vertically like this, then it's gonna grab that thin filament like a magnet. So between this phase and this phase, the process of ATP hydrolysis, splitting ATP into ADP and inorganic phosphate, allows the myosin head to go to the vertical or the cocked position. If you're thinking of this, someone like a trigger, fired, cocked, goes vertical and grabs that thin filament. This is what's known as a cross bridge. So if you can think of maybe a microorganism or an ant or something very small could cross from the thick filament to the thin filament, just walking across this bridge. This is the cross bridge formation where the thick filament has actually made contact with the thin filament via the myosin head. The next phase is what's known as the power stroke, and that's actually the sliding of the filaments or the shortening of the muscle or muscle contraction. All of those mean the same thing, and that's the result of the departure of ADP and that inorganic phosphate. So ADP and inorganic phosphate have taken off, and because they're separating from each other, they're liberating a bunch of energy, and that energy is going to be used to slide the filaments. So we went from this vertical position to this fired position, and the firing, both above and below, is going to allow for muscle contraction. That is the power stroke. After the power stroke, the muscle is in what's known as the rigor state. That is to say, it's in the contracted state, it is shortened, and nothing is happening. Now, in truth, that probably only happens for a very fleeting moment, because once ATP binds to the myosin head detaches and causes that muscle to relax and to return back to its elongated state. Think of this term rigor state and the term rigor mortis. Rigor mortis is the condition that happens 12 to 24 hours after death, where muscles stay in the contracted state. And they stay in the contracted state because there's no more ATP, there's no more oxygen consumption. Without oxygen, our body can't make ATP, so our muscles remain shortened. And that only happens for in a determinate period of time until those muscle proteins start breaking down. But in short,
The rigor state is somewhat a limbo period between the power stroke and detachments, where the muscle is waiting for ATP to bind to the myosin head to cause it to pull off and relax. Keep in mind, when it pulls off, it's still in that fired state. And to be clear, nothing's happening here, nada. And that's because no ATP is bound to that myosin head. Once the ATP binds to the myosin head, we get cross-bridge detachment. So the myosin head comes off, it's still in the fired position, but because it's in the fired position, and that black part of the myosin head is right here, it's not facing the thin filament, it's not grabbing a new G-actin molecule. And that's not gonna happen until ATP hydrolysis happens again, and that's going to cause the myosin heads to go from this fired position to vertical and grab the thin filament. So textbooks may include this in like 6 to 8 steps. I just have it simplified into 4 steps. And one thing I've combined, the myosin head going to the vertical position and grabbing the thin filament all at the same time. So ATP hydrolysis allows for cross-bridge formation. Books may also put the departure of ADP and the departure of inorganic phosphate in 2 different steps. And I'm not saying that's incorrect, but I'm just trying to simplify it and suggest when those entities, ADP and inorganic phosphate, depart, that's going to cause the power stroke to cause the muscle to shorten. And then once again, until ATP binds back to the myosin head, we are in the rigor state, that shortened sustained contraction, which, to be clear, probably happens very, 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 very quickly. And that is the sliding filament theory. More on muscles next video.